Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for UW Extension Cooperative Extension, and on behalf of those folks and our other co-organizers, Wisconsin Public Television, Wisconsin Public Radio, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight it's my pleasure to introduce to you Susan Paskewitz with the Department of Entomology. She was born in Bangor, Maine, and then went to high school in Greenfield, Illinois. She went to Southern Illinois University at Carbondale for her undergraduate and master's degree in zoology. Then she went to the University of Georgia at Athens to get a PhD in zoology. Then she spent several years with the Centers for Disease Control in Atlanta, and in 1991 came here to UW-Madison. She's given several talks here at Wednesday Night at the Lab. She's got an update for us on this new Upper Midwestern Center of Excellence in Vector-Borne Diseases. We get to hear about ticks and mosquitoes and the nasty things they can spread to you and me and our pets and livestock. I'm looking forward to hearing Susan's updates. Please join me in welcoming Susan Paskewitz back to Wednesday Night at the Lab.
Thank you, Tom. Can everybody hear me okay? Is the mic working? Well, it's a pleasure to be back at Wednesday Night at the Lab. I think this is one of those really cool things that we do to promote our science here on campus, and it's going to be a lot of fun for me tonight to get to talk to you about the origin of the brand new Midwest Center of Excellence for Vector-Borne Disease. So I'm going to tell you a little bit of the story about how the center came into being, and then I want to talk about some of the things that we're planning on doing to mosquitoes and ticks over the next few years. So hopefully you'll appreciate that. Before I get too deeply into tonight's topics, though, I would be remiss if I didn't start by saying how much I'm indebted to all of the graduate students and the postdocs who've participated in many of the projects that we have going, as well as, I don't know if you can tell from this, but the undergraduates. We have a huge team of undergraduates every year who work with us out in the field, collecting all those mosquitoes, collecting all those ticks. And then during this time of the year, they're all in the lab extracting DNA and figuring out whether those are infected or not with all the little things that make us sick. So got a great team of people who've done a lot of wonderful work with us. So it's a Center of Excellence for Vector-Borne Disease. So I just wanted to start by making sure that everybody defines vectors in the same way that I do. I'm a medical entomologist, so in my community, when we say vector, what we mean are ticks, mosquitoes, other insects or other arthropods, a mite, for example, that are involved in picking up parasites, bacteria, or viruses and transmitting them. And in the context tonight, I'm mostly going to be talking about diseases of humans, but of course they also do this for animals. So if you treat your dogs for dog heartworm, then you're well aware of the consequences of vectors feeding on animals. So I want to talk about the vectors and the role that they play in transmission of things here locally. But to tell you the story about the Center of Excellence, I actually have to speak a little more globally. Of course, vector-borne diseases are really important in terms of the burden of health in a global context. And so, for example, maybe some of you have heard of river blindness, which is transmitted by black flies, a tiny fly in Africa. And with a lot of exposure, people actually lose their eyesight. And a huge amount of progress has been made in reducing that, the prevalence of that disease. Another one that maybe you've heard of is Chagas disease. Chagas has a lot of impact in the southern part of America and South America, and it's transmitted by a rather large bug called the kissing bug that likes to come out and feed on your mouth or around your eyes while you're sleeping. The bad part about Chagas disease is that it can take a long time for you to develop symptoms, so it could be 20, 30 years after you got infected with the parasite, and then eventually you'll have usually trouble with your heart or trouble with your digestive system that is often fatal. So luckily, we don't have a lot of river blindness or Chagas disease in our part of the world, but probably you've heard of malaria, you know, another vector-borne disease that used to be transmitted here in Wisconsin. So back in the day, there's some wonderful documents from our Wisconsin Historical Society that talk about the history of malaria here in the state. And it was the old forts and outposts where they would report what happened to the soldiers every fall. They called it autumnal fever. Lots of the soldiers would get sick. So we have great records. But of course, now we've essentially eradicated it. We don't have much transmission here unless somebody gets infected someplace else and brings the parasite here and then happens to be fed upon by the only mosquito that can actually transmit that disease. So globally, vector-borne diseases are very important. Of course, there are also a number of things transmitted here in the U.S. that would be more familiar, like West Nile and like Lyme. And I'm going to talk more about those and several others as we go along tonight. But I wanted to use that as kind of a segue into thinking about how what's global and seems maybe not to affect us very much can become local. And Zika was really the story of the year in 2016 for my community, and I'm sure that all of you heard a lot about this virus. It was something that had not occurred in the United States before, had not even occurred in our hemisphere, but it was a virus that had been discovered way back in 1947 in Africa. So it was in Uganda, and it was actually a monkey, not a human, that first showed up infected. And for a long time, very few people seemed to be exposed to this virus. So it wasn't until 2007, I think there were maybe 13, 14 cases that have been documented up until 2007. If there were outbreaks, we didn't know about it, but about that time there was a major outbreak on Yap Island. And then from that, another outbreak in Africa, another outbreak in Polynesia, and then it showed up in Brazil. And that was probably in 2015, perhaps even a little earlier. But by early 2016, it was very obvious that there was a major problem. And it was such an interesting story because Zika is related to other viruses that are transmitted by mosquitoes that we have a lot of information about. So these are things like yellow fever, you probably all heard of, and dengue virus, also very well known. So it's a relative of those viruses, but it was doing things that were very different from anything that we had ever seen in a mosquito-transmitted flavivirus before. In particular, it was sexually transmitted. This was the first time that we saw a mosquito-transmitted virus that could also be transmitted sexually. Just as a story, it was a medical entomologist who made that discovery. He was working in an area where Zika was transmitted. He went home, he gave it to his wife. They figured out that that's what had happened, and then they published a paper on it. You gotta love scientists, right? You get the information out there. So the sexual transmission was one really unusual aspect. The other, of course, was this neurological invasion that caused problems in babies. So we saw microcephaly as well as other kinds of impacts on the nervous system when mothers were infected at certain points in their pregnancy. And we'd not seen that before either, and that, of course, was very concerning. One of the things that happened as a result of this occurring on the scene was UW-Madison became a real leader, and perhaps you've heard from some of the people on campus who did a lot of work on this virus. So we had David O'Connor, for example, and Tom Friedrich and Jorge Osorio all had big research programs up and going pretty quickly to try to figure out some answers. The other place in our state that really got hit hard was our Department of Health Services. So when something like this happens, and the last time it happened was West Nile virus, which came into the state in 2002. When something like this happens, there's a vector-borne disease coordinator downtown, now several of them, but at that time only one, Zip Johnson, and it was Zip's responsibility to figure out how we were going to respond in our state. And so, that's my poor attempt at animation. I couldn't get it to shake because it was the phones ringing off the hook, literally. when this news story happened, she was getting call after call after call from members of the public who were really concerned if they were traveling, first of all, into areas where the virus was being circulated, what was their risk? And then others were very concerned about, what about here? If I go someplace else and come back, or if my neighbor goes someplace and comes back, what are the chances that local mosquitoes are going to pick it up and then transfer it to me? And so she reached out to me and asked me what I thought about this. What did I know about this? We did, ultimately see in Florida and in Texas transmission of Zika virus by local mosquitoes. So that was a result of people going someplace else, coming back infected, then their local mosquitoes fed on them, and then they went off and bit somebody else and transmitted. And we had people who traveled, so we had that part of the story here. By the end of 2016, we had 63 people who were confirmed travelers who came back to Wisconsin with disease, and more in 2017. We also saw another 9 people who showed up. So certainly there's a source, right? There were people coming who could have infected local mosquitoes. So the question for me was, do we have the mosquitoes that could actually do the job? Of course, originally everything was possible. We have a number of different species in the state, and we didn't know whether any of those might be capable of picking up virus and transmitting it. What we did know, though, was that in the places where there was a lot of active transmission going on, so Brazil, for example, it was this mosquito, the yellow fever mosquito that you see right here, that was the main culprit. Yellow fever mosquitoes are interesting. They transmit yellow fever as well as Zika and dengue for a couple of reasons. So there are thousands of different species of mosquito throughout the world, about 180 in North America. This mosquito does occur here in the US, and it is very adapted to people. It really likes to feed on people over almost any other blood source that it could find. It's also very adapted to living right around people and very close association with people. Many of our Wisconsin mosquitoes are not like that. I know sometimes it probably feels like, but honestly, many of them would rather feed on white-tailed deer. They would rather feed on a bird if a bird is accessible. Sometimes you're just in the wrong place at the wrong time. So we thought the Aedes aegypti, or this yellow fever mosquito, was most likely to be the one that would be the problem. That one does not occur here. It's pretty much a tropical mosquito, so where we see it in the US is way down south, Texas, Florida, or places we'd expect to see it, but not here. And I would have been very surprised if we had found it here. The one on the bottom, though, this other one, the Asian tiger mosquito, it's a kind of a secondary vector, and it often will transmit same kinds of viruses as the yellow fever mosquito. Often doesn't do it quite as well, so that's a good thing. You get suppression, but it doesn't mean that it's not able to or can't be an important vector in certain situations. This one has a much wider distribution in the United States. So then the question for me is, do we have that one here? Is the Asian tiger mosquito in Wisconsin. And what you do there, of course, is you go and look at all the records that you have in our museum. We have a Wisconsin insect research collection over in Russell Labs that's full of specimens of all kinds of things, including mosquitoes. We look in the collection, we don't see any. We look at all the records through the years to see if anybody's ever picked it up. Never saw it. In Wisconsin, we do have now, we think, about 60 different species, so it's not just one mosquito. Sometimes people tell me that they think there's only one kind out there, and here, if you look at this picture, you can actually see 2 very different in terms of size, which is sometimes a good tell that you're looking at different species. One of these is actually called the galliniper, and it's huge, absolutely huge, and the other one is a smaller and more avid biter and real common. So I thought, based on the collection records, that it was really likely that we didn't have Asian tiger mosquito in Wisconsin. And I went and looked at what was this older versions of the map that you're looking at here that was produced by Micah Hahn, who actually did her graduate work here at UW and then went out to CDC. And Micah put this together to show where we knew the mosquito had been detected. And so, you know, again, you can see it a little more broadly than you'd expect for the yellow fever mosquito, but not in our own particular area. So look here in Wisconsin, no records ever. But I do want to point out one thing that's interesting when you look at this, and that is look at Indiana and look at Illinois. The red is the places where these mosquitoes are definitely established because they've been detected many times. So what that tells you right away is that there's a problem with the surveillance. It's not a complete record, especially from Illinois. It looks like Indiana's doing a pretty good job, but what we now know is that in Illinois, it's probably established all the way up to Peoria. So if you went and looked, almost all these counties should actually be red. The other thing we know after these last couple of intensive years of looking is notice here, right? And also notice over here. And in both of those places, there's very intense work on mosquitoes. So lots of people looking, doing surveillance. They've got well-funded programs where they know. So I have to say that looking at what was going on in the Chicago area made me a little nervous for southeastern Wisconsin. I was concerned that potentially we had something going on in Milwaukee or south of Milwaukee that we hadn't recognized. We might have an established population and not know it. And I can say that I didn't do enough collecting, haven't done enough collecting there to know whether it was established. So I needed to go mosquito hunting. To do mosquito hunting, you got to know a little bit about what you're looking for. I said there were 60 species in Wisconsin, and they all have really different biologies. They do share some features, though, and probably most of you are aware of this, but I'm always surprised when I go out and talk to the public. Once in a while, I'll find that some people don't know that mosquitoes have an aquatic stage. So the ones that are biting you are almost all the females, and the females are entirely terrestrial. They lay their eggs, and those eggs are always associated with some kind of wet spot, aquatic spot, and the eggs will hatch to produce larvae, and the larvae absolutely require water for their feeding and development. Eventually, the larvae will become a pupal stage, and then the adult will emerge, as you can see right here, from that pupa on the surface of the water and fly off hunting for a blood meal. So if we're going to look for the Asian tiger mosquito, we have to decide if we're going to look for the adults, or if we're going to look for larvae, or we're going to do something else. And we have a lot of different kinds of ways that we could look, a lot of tools. And so if we're going to look for adults, here are some of the traps that we use for that. And so here's one that I use quite a lot. This is called a CDC light trap. It's a CO2 light trap. And in this trap, what you've got is a little light bulb right here, and then you've got a cooler, and we put dry ice in it. And then there's a couple of holes punched, and very slowly over the night, a little bit of CO2 leaks out of those holes from the dry ice subliming. And mosquitoes are looking for a host, and the CO2 in the host's breath is often something that they're focusing on. So they'll come into the trap, there's a little fan then that sucks them down into the cage, and we'll come back the next day and get them. These traps require a battery to run the light and the fan, and they also require carbon dioxide. So you got to have a source of dry ice, and they've got to be primed every day, new bunch of dry ice in, and they cost a lot of money to maintain. There's another trap that we use for looking for West Nile virus vectors. So that's a little mosquito called Culex, or the northern house mosquito. And that particular mosquito, we really like this one because we can collect it very selectively. If we go out with one of those light traps, we're going to get probably 20 to 30 different species trapped in that kind of a trap. In this other trap, what we're doing here is looking for mosquitoes that are trying to find a place to put their eggs. And it turns out that the West Nile virus vector, she, for whatever reason, she likes water that smells horrible. So we make horrible smelling water on the back deck of Russell Labs, just a block or two away. And what we do is we take these big containers, plastic containers, and we put water in them, and then we add manure and grass cuttings and a little bit of yeast, and we cap it and we let it sit there for a week in the sun. And then we come back and we crack the lid, and if it knocks you over, then it's ready. So in this particular trap, then, some of my poor students take gallon milk jugs full of the stinky water, they bait it, and then they set the little trap. And when the mosquitoes come in again, they get sucked in by a fan into a container. That doesn't work for Asian tiger mosquitoes. They don't like that kind of smelly water, so we couldn't use that kind of trap for them. The other thing about Asian tigers is they're daytime feeders. They like to feed, unlike many of our mosquitoes, which are feeding at night or when the sun goes down or when the sun comes up, and not so much during the day. These mosquitoes are really avid during the daytime. So this Lycio-2 trap, it's also not the best for one of these Asian tiger mosquitoes. This trap, this is a sentinel trap, is baited with chemicals that smell like a human. So they put ammonia and lactic acid in the lure, and it works fine for day feeding mosquitoes. In fact, it was developed for yellow fever and Asian tiger. So that's a good one. We have some of those that we use, but unfortunately it's very expensive. So every one of those traps, take a guess how much they cost. It's over $100 per trap. So imagine like how many different places could we even look if it's going to cost us $100 a trap to get them out there. So that wasn't optimal. We also thought about going out to look for the larvae, but larvae are going to be in lots of different places. This is a floodwater species. Here's a ditch where we find some kinds of things. This is the underground catch basin system. You know those grates by the sidewalk where the water flows off the street when it rains really hard? There's a whole system of pipe underground to get rid of all that water so it doesn't flood our streets. Some mosquitoes, especially the West Nile virus vector, really like to breed down there. So you could go there and look for the albopictus, but that's not a place where we would tend to think they are either.
Where were those photos taken?
Well, off the internet. This is a Wisconsin flood here. There's some people who work for Dane County Public Health, and they use a dipper. And this one, I think, is actually ours. This is a slide of ours.
They're all multiple.
Yeah. So what did we end up doing? We didn't end up using any of those methods that I just described that we might use for other mosquitoes. What we did was take advantage of the fact that the Asian tiger mosquito and the yellow fever mosquito are container breeders. So remember that I said that they're really tightly associated with humans. Well, one of the things that they can use is our garbage and debris. And so if we're throwing out bottles, or we've got a bird bath, or we've got an old discarded tire in the woods, or in this case, what you see is a stadium cup, they will lay their eggs in that. And so what we did was buy a lot of stadium cups and a tongue depressor, and then we put water about halfway in, threw in the tongue depressor, thumbtacked it to a tree, and came back in a week and looked to see if any eggs had been laid. If they were, we took it back to the lab and we hatched them, and then we identified the larvae or the adults resulting from that. So we put out, hundreds of these in different places, and what we found, using this ovitrap is what we call it, was, guess what? We hadn't been looking, and indeed we did find it right away. Like within 2 weeks of deploying these traps, we found the Asian tiger mosquito in Wisconsin. And it was in southern Wisconsin. We've not really looked outside of here. We looked a little bit over in La Crosse area, but we focused in the southern tier, so over near Milwaukee, Dane County, Waukesha, a number of different places. But these were the two where we actually found it right away. Our partners in surrounding states in Iowa also ramped up their surveillance. They were finding it in places they'd never seen it before. Similarly, we have partners in Michigan who are finding it, and then those people who really know what they're doing over here in Minnesota, the Metropolitan Mosquito Control District, they reported their 18th find of Asian tiger mosquito in that city. So I don't think they're really widespread or very abundant, but clearly they're being brought into our area with some regularity. You know, the question is whether they're going to be able to make it through the winter. So we have an experiment going on right now where we have eggs out in nature, and we go out every couple of days and we see if they're able to hatch or if they've been destroyed by the temperatures. And so if last week when it was unusually cold here, you were feeling a little annoyed and like you wanted to go to Florida, just think about this, right? This is going to completely knock out the populations, I believe. I don't think we're going to see, at least yet, we're not going to see them make it through the winter. If we had a couple of mild winters in a row, then I think it is possible that we could get them established and starting to reproduce. The other one, excuse me, the other one that I sometimes talk about is the Lone Star tick. And I think I did the last time that I gave this talk. I mentioned that that one has now popped up in a number of places in Wisconsin. But I think as well, that one will not survive a really cold winter. So when we have no snow cover and icy temperatures, that should be good for knocking that one out. That's a relief because a number of people, when they're bitten by that tick, develop a meat allergy to red meat. And of course, that's a huge concern in Wisconsin. The only thing worse, I've told this joke every time I give this talk, but the only thing worse would be developing an allergy to beer or cheese. The other thing to summarize that, what we learned, I think, pretty quickly was that there were some gaps in our knowledge about where these potential vectors of serious diseases that might be imported are, even where they exist. The other thing that happened as a result of the Zika issue was that we, in some parts of the country, realized that we were lacking in expertise in terms of what to do about it. If you did have a problem where you had lots of the vectors and you had transmission, you were going to need to implement some kind of a control program to try to protect the public. In some places where they did that, the people who were implementing the program were not well trained and didn't quite know what they were doing. And in this one case, there was a very negative outcome where a lot of honeybees were killed when they sprayed an aerial pesticide at the wrong time of the day. So that's something that we need to manage a little bit better. And so as a result of that, the CDC recognized through the whole Zika episode that we were lacking information about vectors, that we were lacking trained people who could implement surveillance programs as well as control when needed, and we were lacking medical entomologists, quite frankly. You know, around the country, what we're seeing is a decline in the training of people in the old school kind of medical entomology, where you are going out and collecting and identifying and controlling mosquitoes. What we have now, of course, is a lot of people who are very focused in the laboratory on development of brand new molecular high-tech methods to reduce the disease. That's wonderful too. We have to have that too. But we have lost a lot of expertise in terms of just the basic public health field biology that's necessary when you're dealing with problems like this. And CDC could see that. When they were trying to manage the problem in Florida, so they had active transmission there. You probably remember in Miami area, it was just a mess. And they were trying to get enough people down there to deal with the problem that even I got an email saying, if you can take time off of work and go to Florida to try to help, we really need you. So that was when they realized they needed to work on training. So as a result, they put out a competitive, an application for a competition for applications for the Centers of Excellence for Vector-Borne Disease. And I'm happy and proud to say that Wisconsin houses one of these centers. There were only 5 of them in the country. There were 4 initially, and then they had to add California. But, you know, when we were talking about this, we knew, those in the community knew Florida was going to get one and Texas was going to get one. And we thought New York would probably get one. And we thought California would get one. And it was like, who's going to get the other one, you know, across the country? So it ended up being us. And I'm the co-director with Lyric Bartholomew, so she's pictured here with me. But we have wonderful partners like all around our region. So we have partners in Iowa, in Illinois, in Minnesota, and in Michigan, both at the academic institutions where they can do the research, but also importantly at the state departments of public health. So the health departments, you know, we want to work with them. We want to help them solve their problems, and we want to know what they think their problems are that we could contribute to. So we have great relationships with all of those folks. We were actually supposed to go to Minneapolis tomorrow to meet with the Metropolitan Mosquito Control District and our partner at the health department, but apparently there's going to be some ice and snow, so they warned us off. So part of what we're trying to do with the center is train the next generation of medical entomologists. We want to make sure that we don't lose these really key skill sets. Our center started in January, and by September, we were already seeing some of the fruits of what we did. When the Hurricane Harvey came through Texas, the flooding as a result of that hurricane was enormous, and within a very short period of time, there were millions of mosquitoes. So that could cause two problems. One problem, of course, is they might transmit disease depending on which mosquitoes they are, so that would have been a disaster. The other thing is they can really hamper the recovery efforts. So imagine if you're out there and you're trying to clear debris or you're trying to build or repair and you're being swarmed by lots of mosquitoes. It's just a huge mess when these kinds of flood events happen. And they couldn't handle it, actually. So they had a company come in and we're working with them. The company got overwhelmed. And so the next thing that happened was they reached out to us and said, do you have anybody who could come down and help us? Because what we need to do is ID these mosquitoes, count them, and figure out whether our control measures are working, and then also help us with an assessment of whether the disease vectors are beginning to rise. So we had two great students who went down there as Melissa and Aaron, and they had a wonderful time and came back with some great stories, and they contributed a lot to that program. So Zika, I think, is the reason why the center was established, but of course there are lots of other mosquito and tick-transmitted diseases in Wisconsin, in our region, that we have to worry about. And so West Nile is one that you probably all have heard of. Lacrosse virus is the major cause of pediatric encephalitis in the country. One that you probably have never heard of is Jamestown Canyon virus, but we've been seeing quite an increase in human incidence of this particular viral encephalitis, neurological disease, over the last few years, and we don't understand why. And interestingly, we only know that too because of this regional center. So we've been talking a lot to each other, and we began to realize that in Minnesota and Wisconsin, this unusual pattern is showing up. And so we're going to be doing some work to try to understand what's happening with JCV. So part of our work is to discover, respond when emerging things like JCV or Zika or West Nile happen. Part of our work is to look for new control strategies. And so one of the people in my laboratory now is working on bacterially based repellent. So one of the things you can do to protect yourself if there's a mosquito problem is you can use a repellent on your skin, right? We would call those topical repellents. Many people know about DEET. That's the one that would be made by our own SC Johnson, you know, here in Wisconsin, in their off product. And it works really well, but a lot of people don't like to use it. And so we've been looking for some alternatives, and we've got something that actually works really well. It looks like it keeps the mosquitoes from feeding. So that's one of our strategies that we're going to pursue. We're also really interested in assessing how well our current strategies for controlling mosquitoes are working, and also for surveying them. And then thinking a little bit more about the human aspects here. I'm always very targeted on the vectors and what we can do to the vectors, but I think we need to think a little bit more too about how we're interacting and whether there are some things that we can do that would reduce those interactions. So, you know, I think a core tenet for our center is to think about how we can reduce the risk and prevent the disease, but at the same time protect the environment. So that disaster with the bees, that's not something that we would want to repeat around here. And to the best of our ability, actually, we don't want to do any aerial spraying for adults at all. You know, we would do that, I think, if we were having an epidemic and an outbreak and had no other alternatives, but we would prefer not to get to that point. So thinking about how can you control mosquitoes if you're not going to spray the environment gets you back to thinking about trying to control the larval stages, you know, get them in the water. And we've been working for a number of years now on trying to think about how you can do that biologically. So where and when can you introduce fish, which are really good at eating mosquitoes in some environments. We also have been working on this little thing, which maybe some of you don't know about. It's a microcrustacean called a copepod. And if you've heard of copepods, fine, but you still will not have realized that some copepods are predators, and they will actually eat those tiny little mosquitoes when they hatch out of those eggs. So we've been working on trying to grow these tiny little copepods and see if we could release those into the environment to reduce the larval stages. Another thing that we do that I think is really important is to test different methods that are out there for the general public to use to protect themselves. And these might be things that you'd put on your property, or they might be things like topical repellents like DEET or other things that are available. So we test them, or if we don't test them, I go out and I find the science and then I summarize it, and I put it on my webpage, which you can find if you just Google search Wisconsin mosquitoes. And this will come up. And what you'll see here is there's a section that names all the mosquitoes that we're aware of in Wisconsin, tells you a little bit about their biology. But then this section on preventing bites has a lot of information on what things work and what things don't, at least so far as we know. So as an example, if you ever wondered whether putting purple martin houses out in your yard, which many people think is a useful tool for reducing mosquito populations, if you were curious about the evidence, you could go to this website and take a look and see what that evidence is. And what I can say is that evidence is, there is no impact. And it's all based on some really interesting old work where somebody speculated on how many mosquitoes it would take for a purple martin to survive. That was all. It wasn't even an experiment. So many other interesting things here if you want to go and take a look, and if you ever have a question about whether these might work or not. The other thing on the mosquito website that if you're in Dane County might be interesting to you is every summer we put up our tallies, and we try to do this on a weekly basis for our surveillance for mosquitoes in the area. And we'll also, I think, here report what we're seeing with West Nile virus. This is interesting because you see those peaks. I don't know if you can make out the colors there, but last year was the most mosquitoes we have ever seen in the 5 or 6 years that we've been tracking them. It was really amazing. Tens of thousands came in. You can pity the poor undergraduate who had to sit at the microscope, sort all those out, identify every one of them, count them, and tell us what was out there. So it was a really big year, and this is a place every year where you can go to see kind of the timing and what's happening. All right, so I know for many of you, you're probably not convinced that mosquitoes are the most interesting thing that a medical entomologist could work on. You're wondering, what about ticks? And ticks, of course, are very important in Wisconsin. They are involved in transmission of Lyme disease, which is a tick-transmitted bacteria. And there you see one of those Department of Health Services maps that shows you the incidence for Lyme disease cases in our state for 2016. So we have a lot of Lyme. It's not just in the northwestern part of the state. It's really, you could pick up one of these ticks anywhere the habitat is right. And one of the reasons that we spend time on the tick-transmitted diseases too, I think, is because so much change has happened. So when I first got here, it was a very different story compared to what we see now. And this was a recent paper that came out by a colleague of mine who's at the Centers for Disease Control. And she looked at all the records for the deer tick between 1907 and 1996. So these were all the counties in red where there was an established population, and in blue where there had at least been a collection, a record of a tick, and in white probably not much information. But what you could see from this was a huge change from just 1996 to 2015, where basically I would say now our entire state is infested. Any place in the state where you go, where the habitat is right, these ticks can be present. I don't think the last few times we've been out, we've been able to find any places where they're not present. So they are very widespread. You also see here, if you're in Michigan, you've got a looming problem. You can see a huge change in Minnesota. You know, a lot of expansion of the tick there as well. You know, in Wisconsin, along with the expansion of the tick range, we've also seen this skyrocketing number of Lyme disease cases in the population. So from about 400, 500 a year when I first got here, last few years, the confirmed reported cases, those are only the ones that get to the state, and the vast majority never even make it to the state. The confirmed reported cases is 3 times higher than it was just a couple of decades ago, so 1,500 cases. CDC would say that that's almost certainly a huge underestimate, maybe by as much as 10-fold, so maybe more likely to be 30,000 cases a year. It's huge.
The previous slide, the number didn't make any. Should that be 1907 to 2015 or 2000? Not 1997 to 2015.
What she did here was she combined all the dates in the first map, and then in the second she just added everything that had been found since 1996, but kept the base map. So lots of Lyme disease. The other interesting thing I think about Wisconsin too is that we've become kind of a hot spot for newly emerging tick-borne disease. And so you can see Anaplasma phagocytophilum was first discovered here in the early 1990s, and I remember working with the doctor from the Duluth clinic, Johann Bakken, who found this in patients that were showing up in his office from both northern Minnesota and northern Wisconsin. In the last, 5 years or so, 5 to 10 years, we've also seen a new kind of ehrlichiosis, another Borrelia pathogen, not Burgdorferi, which is the classic Lyme disease pathogen, but one called now Borrelia mayonii has started showing up in our citizens and causing a Lyme-like syndrome. Just last year, I think, for the first time, Borrelia miyamotoi, which is causes a relapsing fever kind of a disease. So it's not like Lyme disease, it's a different kind of illness. And then for the very first time this year in 2017, Babesia divergens. That was in Michigan actually, but it's close enough for me to think that it's probably circulating here as well. So lots of other kinds of diseases, all actually being transmitted by the same tick. So the deer tick is the culprit here. I don't want to alarm you because most of those are very rare. We do not see a lot of human cases of those diseases. A few more of Babesia, for example. But this Anaplasma, we definitely see quite a bit of that with 400, 500, 600 cases every year. Again, that's what get reported to the state. It's not necessarily the actual number of people who are getting sick. So we do a number of things to try to help because of the tick problem here. One of the things, again, is educating the public. And I started this website a number of years ago because we were hearing from people in the state, especially in the southeastern part, it seemed, that they would go to their doctor and they would have symptoms like Lyme disease, and the doctor would tell them that they couldn't have Lyme disease because the ticks weren't there. And that reflected what the case would have been, 20-odd years before that. It was true that we just didn't see many ticks in eastern Wisconsin. But that has changed a lot, as I showed you. And I put this up so that people would be able to go here and look and see what's the distribution of the ticks and show it to their healthcare providers, and also to have information about the prevalence of all these different agents in the ticks from different areas. So there's a lot of maps in here that will show you how many ticks are infected with which parasite from what area. One of the things that I want to do here, I haven't done this yet, but now we're going to expand this a bit, is to do the same thing that I have on the mosquito site and talk about what can you do to protect yourself, what works and what doesn't work. And so we'll be adding that over the next year. And then I'd like you to stay alert this year because we're also excited about this. Coming soon, we're hoping to have a way for you to take a picture of a tick and send it to us so that we can give you an identification and then some information about what you ought to do. What we've come to realize is that although many people are really good at identifying deer ticks, some people are not. Some people will, I've had people send me pictures of pseudoscorpions. And they were crawling on the curtains and they thought they were ticks. So, you know, we do have several other kinds of ticks that you could get on your person. The wood tick in particular would be one. And people sometimes even confuse that one with deer ticks. So we'd really like to have this system where you could send us pictures. We would ID it. We would tell you, you know, what's our advice for what you'd need to do next. We're going to model it under this. This is the Tick Spotter app out of the Rhode Island Tick Encounter Resource Center. If we don't get ours up in time, you can definitely use theirs, though. Go ahead. And then, let's see, the other thing I wanted to talk about for the next couple of slides is some of the work that we've been doing in terms of control and what you can do to reduce the risk of tick-borne disease, given that now you can be infected with any of this suite of 7-ish different parasites if you're bitten by a tick. See, I don't have Borrelia burgdorferi. Oh, yes, I do. Not Borrelia mayonii, I don't have up there. So if we're going to think about control and what we can do with these ticks, I wanted to just start first and mention, you know, a little bit about their life cycle. So they start life as a larva, that little tiny 6-legged stage that you see there. If a larva is successful at getting a blood meal, it'll molt to the next stage and become the nymph. The nymph, if it gets a blood meal, will become the adult, either male or female, and then those, of course, will mate and produce the eggs in the next generation. We think it's the nymphs that do most of the disease transmission to humans because they're so tiny. It's not that the adults couldn't transmit, but usually you feel them and you get them off in time. The nymphs, though, if you look at the fingernail there, you see just how tiny the nymph is in comparison to the adult. And many times I have been in the field doing collections. I am very conscientious about doing my tick checks because I don't want to get sick. And still, I'll do my check. I'll be sitting, talking to my husband. I'll look down, and there's a tick crawling up my knee. You know, there'll be one of my moles will get up and start walking around. So the nymphs are just very hard to manage. If we're thinking again about control, one of the important things to recognize is where we might want to do it. And so if you live in a big house on a big open hill in a meadow or a cornfield that just got turned into a subdivision, you probably don't have a lot to worry about. Those are not the kind of places where deer ticks thrive. Where they do thrive is in woods. They really require high humidity, some shade, woods, and the animals that inhabit those areas to do well. And so here you can see a map that overlays the Wisconsin landscape types with the distribution or the incidence of Lyme disease. And you see how well it matches where the forests are. That tends to be where people are getting exposed to this pathogen. I mentioned already that ticks need to take a blood meal. That's all they feed on. That's really important for them to be successful. Here's a picture of what happens when they do feed successfully. So it takes them about a week if they're an adult, and it goes from that day 1 to day 7. They're going to put on 200 to 600 times in weight over that period. So you got to picture me expanding from my 100 and something pounds by a factor of 200 in 1 week, and think about the physiological challenges that would involve, right? My skin's going to have to really expand and grow. And blood turns out to be a very nasty substance. There's a lot of heme in it, a chemical that's really hard for organisms to manage. So they have to have all kind of detoxification mechanisms and other things to be able to live this life. In order to get the blood, the adults usually like to feed on larger animals, so they'll feed on you, they'll feed on your dog. They really like white-tailed deer. That's a favorite host for them. We never see them on small things, though. Instead, on the little things, the little rodents in the environment in particular, it's the immature stages. The larvae and the nymphs like to feed on the little guys. And in our state in particular, the white-footed mouse and the deer mouse, so a couple of species, are very important as reservoirs and in terms of feeding ticks. And also chipmunks are likely to be a really important part of the cycle here as well. So with all of that background, we can think a little bit about what's the toolbox or what could be the toolbox for doing some control ourselves. And this was something that I took from Ben Beard, who's at the Centers for Disease Control, and he had listed in white here all the different things that you could do, and in yellow all the things that people are working on that we would hope to have available at some time in the future. So you can see there are things that that people can do. You can avoid tick habitat. Make sure you build your house in that cornfield. You can also think about other things that we could do to people, like it'd be really nice if we had a vaccine that was effective. But there are things that you can do that would involve also manipulation of your habitat, so your landscape perhaps around your house, or thinking a little bit about how we might manage the ticks that are out there in the environment. Here, where's my, here we go. Like trying to use some kind of a pesticide, but hopefully something that's a naturally based pesticide rather than a synthetic. Or, and this is a really interesting aspect, when you've got animals involved in the disease cycle, you can think about trying to target the animals. And so this is where we've been putting some of our efforts in the last few years. There is a system where you can apply a chemical to mice and it gets on their fur, just like if you were treating your dog. So, you know, you put the front line on the back of the dog if you have pets. This is what we're trying to do with mice so that any ticks that get on the mouse then get a dose of the front line like chemical and they don't survive. And the same thing people have tried for deer, but unfortunately to do it, it works with deer, but we've got CWD, which makes it a little bit harder to bait deer into a place where they could get treated in that way. But they use it on the East Coast in some areas. So one of the things that we are trying to do right now is to assess all of these different tools that you could use and again provide you information about what will work, what we think will work best. So is it going to be landscape management? Is that what we would think you should put your time into? Or should you try these little treatments for the mice? So we've had a study going for 4 years now in the UW Arboretum, where we've been looking at trying to do an integrated management program where we hit them in 2 different ways. And so in that program, what we're trying to do is change the landscape so it's not as good for the ticks. And we're also trying to treat them with the chemical that will kill all the ticks. And so we're using, I think this might be, no, wait a minute, got it on the next one. Here, what we decided to do in the Arboretum was to remove buckthorn. In the US, there's a number of studies in different places that have shown a relationship between invasive shrubs and tick density. And I think probably they get very dense. They provide great harborage for the mice and a place that maybe also attracts the deer. And so you just get a real intensity in association with those shrubs. In the Arboretum, we had some preliminary information that indicated that the more buckthorn you had, the more ticks you had. So we thought that that would be a good place to start, would be by removing that to see what would happen. The other thing that we tried to do was this targeting of the mice. And so this is our tick tube study. So you can buy tick tubes yourselves. And what you do is you've got this little cotton. It's been treated with a chemical called permethrin. We put them in the PVC pipes. We throw them out where we think the mice will go. The mice come and get the cotton in this case, and they make their nest. They line their burrows with it. Then every time they go into the burrow, they get a little swipe of the stuff on their fur, and when the ticks get on them, then it kills them. So we go in after we've deployed these different techniques, And we sample for ticks that are in the environment looking for a host with this dragging method. You see the white blanket there. We also trap the mice. We do live trapping of the mice to see how they're doing, if they have ticks on them or not. And what I can tell you is we have enough data now, I think, to say that the cotton ball treatment, the nest material, actually did reduce the abundance of the ticks in the environment, those that would be looking to feed on you, the density of those nymphs. It did successfully do that. So when we put the cotton out in 2014, those mice probably killed all the larval ticks that fed on them, and that meant that the next year, when the nymphs were out looking for a host, they were seriously reduced in the population. And we've seen that now for 3 years running. So I think that that's real. Sadly, though, the buckthorn didn't work as well. When we removed the buckthorn, it looked like we had a significant impact the first year. So then we did see a reduction, but after that, nothing. So I'm not sure that we've changed the landscape enough to really make a difference in that context. So ultimately, what we're going for here, what we're trying to do is to come up with a sort of a do-it-yourself toolkit for homeowners and or for communities. So we right now have a couple of communities that have come to us and asked for some help, where a number of people in these neighborhoods all want to work together to implement methods that hopefully will reduce their risk and their grandkids' risk. And we want to be able to provide advice about what should you do in the Wisconsin context, which might be different from what you do in the East Coast. So we'll be continuing to develop these studies over the next couple of years.
What are those cylindrical objects in the picture?
Those are the ones you can buy, the tick tubes that you can buy. So you can go to Thermacell, or there's another company called Tick Tubes, I think. If you just Google that, you're going to find them, and you can purchase for deployment. And then the very last thing, I would be remiss if I didn't mention this. My postdoc would kill me. She is working on a new tick app. And what she's trying to do with this is to get more information about where you're actually picking up the ticks. We feel like that's a big piece of the story that we don't fully understand. So are you getting it when you're going fishing? Or are you picking it up when you're out in your garden? Or is it when you're actually holding your cat who's been outside and maybe picked up the tick and it transferred to you? Or something else where we could actually change our educational methods and be a lot more targeted. So we're going to have people enrolling this year, and you'll be asked to fill out a survey initially, and then the key thing we're hoping is that you will agree to allow us during the peak of the tick season to ask you to fill out a survey every day about whether you found a tick on your person or not, and then what you think you were doing when you actually contacted that tick. In the future, after we get through this first year of trying to figure out what the risk factors are, we're also thinking about whether this app might be a valuable tool in terms of disease control. If we just could send you a reminder every day, during the peak season in June, or maybe a couple of weeks in June, to say, did you do your tick check today? And if we could get that kind of a message out there routinely, if you'd agree to it, then we would do a test to see if it actually makes any difference in terms of the incidence in Lyme disease in people who are using the app. So I'm going to finish up here by just recognizing the agencies that have provided support for some of our work. So our Department of Health Services downtown has been a wonderful partner and has often paid for undergrads to go out and collect ticks in the summer. The USDA supported the Arboretum project for the last 4 years. The CDC with our new big project, which I think is going to bear a lot of interesting fruit in the next few years, as well as several other federal agencies. And I'm going to end here with a picture of my field team heading off into the woods. And I just want to make sure that you notice, just in case he ever sees this, I want to make sure you notice this guy with the pink pack with the brats on it. That's the punishment pack. If you show up for work and you forgot your backpack, you got to carry. So I thank you for your attention. Okay, so I think I'm ready for questions now. Yes, sir.
I was wondering, although you didn't mention it in this presentation, do you have any, or have you done any research into the question of flea-borne diseases, for example, like bubonic or pneumonic plague? I know that those are an issue because several western states are considered to be permanent plague reservoirs.
Correct, yes. So he's asking whether we've ever done any research on the plague and the fleas that are important as vectors of plague. There is actually right now a major outbreak of bubonic plague in Madagascar. And here in Madison, we have the National Wildlife Health Lab, and there's a researcher out there, Tony Rocky, who does a lot of work with plague. In fact, I just hired a graduate student who just completed her work with Tony on plague to come work with me on ticks. I also have another student who's doing some work with on plague in the laboratory. So we have a new flea culture going. I'm not going to be doing a lot of that myself, but I'm hosting them. So I hope to learn a lot more about what they're doing.
Oh, great. I had a quick follow-up, too.
Sure.
First of all, where is Jamestown Canyon? And also, what is the definition of a riverine forest?
Yeah, I don't know where Jamestown Canyon is, so that would be the place where they first identified the virus, actually. But so he's asking what is Jamestown Canyon? Where is Jamestown Canyon, for which the virus is named? And just like with our lacrosse virus, that was the first place it was ever identified, so it was named for the location. I'm not sure about JCV. And the other question is, what's a riverine forest? A riverine habitat for us is usually the corridor alongside a river. So, it can extend however far the wetlands and the lowlands are alongside that river.
Okay, because I noticed on your map it appeared to be confined to the area around Cairo, Illinois.
I'll have to look, I'm not sure. Yeah. Yes?
Thank you. Very engaging. I understand that Jorge Osorio has been working on the Wolbachia bacteria and its ability to interrupt the transmittability of the Zika virus. And they're thinking it might work with dengue. What's the status of that research?
So she's asking about this amazing new tool that's been developed for trying to interrupt transmission of viruses in the mosquito. It's called Wolbachia. It's a symbiotic bacteria. And people have spent a long time trying to figure out how to move these Wolbachia around, because they usually tend to be quite specific for a particular insect that they reside in. So 2 different kinds of Wolbachia, one from a Culex mosquito and one from Drosophila melanogaster, have been moved into Aedes aegypti. And when they put it in there, what they thought they were going to find was that infection with Wolbachia would shorten the lifespan of the mosquito sufficiently so the virus and other pathogens wouldn't be able to complete their life cycle. So it just would make it so they couldn't transmit. What they actually found in some cases was when you put these bacterial symbionts in, all of a sudden the mosquitoes could not transmit the virus anymore. They didn't get infected with it and they didn't transmit it. It didn't really do the lifespan thing so much, but it interfered with the ability of the virus to colonize the mosquito. So there have been actually a number of studies now where we're releasing it, where people have grown these things up in sufficient numbers. They're infected with Wolbachia, WMEL, and the other one in Australia, actually, and releasing them in large numbers and then looking to see what that does to transmission. So the last time I talked to Jorge, where they were releasing it in Colombia, actually, I don't think that they had data yet on whether it suppressed disease in the mosquito or in the human population in that study. Sure. But isn't that an amazing tool? It's like the the genetically modified mosquitoes that people are thinking about trying to release in Florida. That was quite controversial, obviously, but yeah, similar kinds of those are the high-tech bench studies that we need to do. Yes?
How will they be recruiting for that new app for the ticks?
How will we recruit for the new app for the ticks? Yes. Mm-hmm. So you saw My Wisconsin Ticks website. And then we also have a website for the Midwest Center of Excellence for Vector-Borne Disease. It'll be in both of those places for sure. We also, with the communities that we're working with, we're going to go out and directly ask the homeowners and the people who have been interested in the tick control projects if they would like to participate. We're kind of hoping for maybe 1,000 people, but I think when they piloted this last year, a version of it someplace else, they ended up with 12, which was an insufficient sample size for what we were trying to do. So if you have suggestions for how we can publicize it more, let me know. That would be something that we're really interested in figuring out how to get it out there to the greatest population.
Your website again is the
Wisconsin Ticks. We'll have it there, and it'll probably be on the front page. So as soon as you go in, you'll see the link.
Okay.
It's not up yet.
What's the time frame for that?
Well, we want to get it rolled out before the season, so we're really hoping to have it ready in May.
Okay.
Yes.
What do you think of I'm sure I saw it maybe in Science News that the outbreak in Brazil coincided with a redefinition of microcephaly.
Microcephaly?
Yes, and that really there aren't any more cases of it. It's just that they redefined what a small head was considered.
I didn't see that. What I have seen, so I can't speak directly to that. What I have seen is the laboratory studies using the primates that have been able to replicate what they see when they infect the primates with the viruses. They're able to see those kind of impacts on the nervous system of the offspring from those organisms. So that seemed like a pretty tight connection to me. But yeah, so she was asking for the viewers about a recent study that she read in Science News that suggested the microcephaly might have been a false relationship because it was a result of a redefinition of what microcephaly was right around the time when the outbreak happened in Brazil. So, and I'm sorry, but I can't speak to that.
And is that the one that came out of UW-Madison here? Report?
I think so, yeah.
Yes?
For Lyme disease, is it the deer that carries the basic disease?
So the question is for Lyme, is it the deer that are the major reservoir that are carrying the disease? It's not. So what we know is an interesting thing, and that is that deer, although they're getting injected all the time because of the feeding of the adult ticks, they don't get infected with Lyme. There's something about their immune system. It's a part of their system called the complement cascade in the bloodstream that is able to kill the spirochetes. So when they get infected, they just wipe it out. The major reservoir, the way that the ticks get infected, is when the larvae are feeding on small mammals primarily. So that's going to be chipmunks, it could be, or these little white mice, white-footed mice, the deer mice, shrews. Some of the shrews can actually do it. Some birds can do it.
But it doesn't pass in their gender. Can't pass it on to its young.
Great question. Yes. That's true. So he's asking whether a mother tick could pass it on to her eggs and her offspring, the Lyme disease spirochete in particular. As far as we know, that doesn't happen. There are, though, some other pathogens, and one of them I mentioned, this relapsing fever Borrelia. That one can, in fact, be transmitted from the mother onto her eggs. So we would describe that as transovarial. That means that the larvae can also be infected. I don't see a lot of people being bitten by larvae or reporting being bitten by larvae, so I don't know how frequently that happens, but it is a possibility. Yes?
This is more related to public policy. As I remember, when the most recent outbreak of Ebola was still in Africa, the health authorities in this country were using the explanation that don't worry, it just can't happen here. They never gave a reason why that would be, but they just kept saying it just can't happen here. And as we know, that wasn't exactly correct. So I guess my question is, with regard to the types of pests that you're researching, or any other potentially dangerous outbreak of disease, do you think that the mentality among the public health authorities is still very much this one of it just can't happen here, so don't worry about it?
So he's asking whether I think that the mentality amongst the public health community is that these disease outbreaks like Ebola or other things are unlikely to happen here, so we don't need to worry about them too much. Well, I would say in my own community that works on the vector-borne disease, I don't think that that's true at all. And I think that West Nile was a wake-up call, so when that happened, you know, there was a lot of confusion originally about what that was. When that happened in New York in the late 1990s, it was originally thought to be something called St. Louis encephalitis, which we'd known about in the U.S. for a long time. So they got it wrong, but then they realized it, you know, it was behaving oddly. It was killing birds, which shouldn't have happened if it was St. Louis. So that was kind of a wake-up call. And so I don't think that when Zika started to happen this time around that anybody was really, in the public health community anyway, was thinking that it couldn't become a thing here. We had seen dengue virus in Florida, so we knew that the right vector and the possibility for transmission could happen there. So, yeah. Yes?
So how research regarding ticks, how different is it like in Europe and other countries compared to the U.S.?
So asking about the research related to ticks or more about the disease transmission in Europe? Yeah.
That's correct.
So Europe has similar kinds of Lyme disease pathogens, Burgdorferi, as well as a couple of others that so far we haven't seen show up here in the United States. And they have a tick-borne encephalitis, which I think is like our Powassan virus. They're kind of related. You know, again, we don't tend to see a lot of people getting infected with the encephalitis virus. You know, in terms of Europe, I don't think they have the same sort of Lyme disease incidence that we see. It does seem to be, they have it, but I don't think that the numbers in the population are similar to what we see here at all. So, and it does keep spreading and popping up in new areas, but.
Are the ticks that infect domestic sheep and goats at all related to the deer tick or other human disease transmitted ticks?
She's asking if the ticks that infect domestic goats and sheep here in Wisconsin.
Well, in North America.
In North America, if those are deer ticks or something else. I think you can get other things on them. I'm just not aware of reports. I'm trying to think about our own work of whether I have anything that would suggest Ixodes scapularis. Well, maybe. So we've done this one project that we call the Survey of Wisconsin Animals for Ticks. It's a SWAT program. No goats have come in, unfortunately, so I can't tell you results from that. But we have gotten results from elk, from deer, from raccoons, from possums, lots of smaller things. And Ixodes scapularis, the deer tick, is on almost all of that. Now, not always every stage, right? So the adult's not on the small things. But so I'm going to guess that yes, I don't see a reason why, if they're on that broad an array, that you wouldn't get deer ticks on goats and sheep as well.
Because sheep shearers find ticks.
Ah, okay. Next, do you shear sheep?
No, I don't.
Okay, well, if anyone out there shears sheep and you find ticks on them, we'd be happy to diagnose them for you. So Tom is pointing to a question in the back. Yes?
If the deer do not get Lyme disease, are there studies going on what might be in the composition of the deer that would repel the Lyme disease?
So she's asking if deers don't get Lyme, are the studies going on to figure out why? And I think that, from my understanding of the literature, we do already know that it's this complicated system in your bloodstream called the complement cascade. It's part of your innate immune system, and there are specific molecules involved in that cascade that are able to bind to and kind of tag the spirochete in a deer blood, but it doesn't work the same way in our blood. And I'm not sure why it doesn't work the same way in our blood. There must just be some subtle molecular differences between, because I think the systems themselves are quite similar. Do you have a question, Tom?
Yeah, would you like to speculate on why so many vector-borne diseases are found here in Wisconsin so early?
So Tom is asking me why we see so many emergent tick-borne diseases here in Wisconsin, which is an excellent question. And I do think that there's a reason for that. I think it's probably, it's a couple of things. One of those things is that a lot of people who get diagnosed in northwest Wisconsin with disease end up working with the Mayo Clinic in some kind of a way. And there is a diagnostician there, Dr. Bobby Pritt, and Dr. Pritt is really good at what she does. And when she sees something that doesn't look right, she doesn't let go until she figures out what it is. And so that is the reason, I think, why Ehrlichia murus, for example, the Borrelia mayonii, a number of the most recent new things have been identified is because of Dr. Pritt's work. I also think, let me tell you one other thing that I think is interesting, and that is that our ticks here are not the same as the ticks on the East Coast in terms of their behavior. So on the East Coast, what you tend to see is in June, you get a peak of the nymphal stage. So they're coming out after the winter, they're out there feeding, they get on the mice, they infect the mice. The larval peak doesn't happen until August. So if a mouse gets infected in June, that's got to stay infected until August in order to infect the next year's cohort of larvae, right? So that's going to select for pathogens that are persistent in the reservoirs. In our case, what happens is nymphs and larvae, they come out at the same time of the year. We tend to see them peaking in June to early July. So anything that's in those animals that's a short-lived kind of pathogen that maybe isn't very infectious for very long, it's still going to be able to infect the larvae that are co-feeding on those hosts at the same time. So that may be contributing to emergence of some of these things that perhaps wouldn't if we had that more asynchronous kind of a phenology. Yes, sir?
Yeah, I'm curious, and I don't know if you know about this, it's more East Coast, but a friend of mine who lives in New York State who got Lyme disease and ended up with a pacemaker because of it. This is a number of years ago. When he went to his doctorate, the mark he found, the doctor diagnosed it as a spider bite, and so it didn't treat for Lyme, and later he developed Lyme. Have the techniques in terms of distinguishing Lyme disease tick-carrying bites gotten better? So that a doctor would know that right away?
I don't think so. So he's asking whether the techniques specifically for identifying something as a tick bite rather than some other kind of a bite, like a spider, have gotten better. I don't think so. I don't think it's particularly obvious. You know, if you were spider bite, or I've seen people who are convinced they've had a spider bite because they've got a little ulcer around the spot. It ends up not being spider bite at all. It ends up being MRSA with the methicillin-resistant Staph aureus that they must have got a little poke in their skin and then got exposed in the environment. So a lot of confusion about what those are, and I don't think it'd be easy myself. Yes?
Based on what we were talking about, Mayo and different doctors seeing looking and trying to root out these things, these symptoms and stuff. You may have heard of the ILADS group, so does that mean that some of these doctors are colluding and seeing symptoms? This is based on his question, seeing more of the same types of symptoms that help them lead to a diagnosis and the type of testing they may need, because a lot of the testing is very specific. For instance, Babesia is in the blood supply, Red Cross admits it, but it's hard to test for. That's why we've heard of amber. They found a spirochete in a tick that was 5 million years old. It's always been there, but were we looking for it? And you conduct with a lot of your research and stuff that you're doing.
Yeah.
So is symptoms
coming
more into play these days?
So I think, you know, I work on the tick end of things. I don't really work on the diagnostics or the things that the physicians and the clinicians are responsible for identifying and properly diagnosing. I know that for Lyme, there's a terrible problem with the earliest stages after infection. You know, you can come in, see your doctor, have non-specific symptoms. If you get the EM rash, you're actually lucky, because if you get the EM rash, very clear, easy to identify.
Is that because of a slow replication rate?
Is what?
Slow replication rate of a parasite?
The reason why you get hard to diagnose in the early stages. It could be, but I was actually thinking more that it's because of the slowness of your immune system in responding. You know, you don't make antibodies like instantly when you've been exposed to something.
Immune system if they're going to survive.
So I'm thinking about like when you get infected with Lyme disease to start with, you know, those first few weeks after you're infected, you don't really have a lot of antibody, and antibody is primarily what people use to diagnose, you know, that plus symptoms, right? So you go in with symptoms, but they're maybe kind of nonspecific. You've got fever, for example, general malaise. It's in the summertime, so if my students come to me and that's the kind of symptoms they have, I always do tell them, go see the doctor, even if you didn't get a rash. You know, you should talk about the fact that you've been exposed, and summer flu is unusual. It'd be worth your while to get a prophylactic treatment, get some kind of an antibiotic treatment.
A couple more, one back there, one up here.
Sure, I'll take one up back here.
On one slide that you had, said the first case of Lyme was identified in Wisconsin in 1969. What was the slide?
Correct, yes.
But I thought it was first identified in Lyme, Connecticut.
Yes. Okay, so he's asking about my slide that identified the first case of Lyme in Wisconsin in 1969 versus what we know about Old Lyme, Connecticut, which is where the disease gets its name. And so what we can say is that when that case was written up by a doctor named Scrimenti, and he published it in 1970. He didn't call it Lyme disease, but it really clearly was Lyme disease. It was a erythema migrans with all of the constitutional symptoms that you would expect to be associated. The EM rash was very definitive in that case. So he didn't call it Lyme, but it was, people generally agree that that was the first one in the literature. Old Lyme, where it actually got its name, I think is also a very interesting story because that was the result of mothers in a neighborhood recognizing that too many of the kids in the neighborhood were getting rheumatoid arthritis. And that shouldn't have happened. They recognized that that was a cluster, something unusual about it. The incidence rate was very high. And they went and sought additional help to figure out what the cause was. And then did. You know, it took a few years, but they were able to identify both the pathogen as well as the tick involvement in transmission, and hence they named it Old Lyme. I tried to argue for Borrelia mayonii being called something like Borrelia wisconsini, but I didn't get anywhere with that. Maybe the next one. Yes?
Is there something going on in the immune systems of the ticks and mosquitoes that makes them more susceptible to disease, or is that maybe just we're identifying the diseases more now, or the populations are increasing?
Oh, that's a really good question. So she's asking whether there might be something changing about the immune system of the mosquitoes or of the ticks that might be allowing them to be more tolerant and, you know, more infected than by all these different pathogens. You know, we know a lot about the mosquito immune system. We know a lot less about tick immune systems. I do know that when they're stressed with sublethal doses of insecticides, it changes their immune response in the mosquitoes. But I think, you know, for the most part here, they're probably not getting dosed very much. There's not very much active mosquito control in Wisconsin. So I don't know. It's an interesting hypothesis, but I don't think we have any data that would suggest that that's happening. Yeah. Yes. One more.
Do you think that climate change and the warming we've been experiencing is having an impact on the range of these vectors?
Well, what we know about, she's asking about climate change and the range of our vectors. What we do know is that the deer tick is moving into Canada, and they do think that that's a result of climate change. It's showing up in places where it never did before. It's also expanding south, so that's probably not a warming, you know, altogether. The range isn't just because of warming. In the case of the mosquitoes, I don't know whether I could say that for right now, but I do think that probably within 30 to 40 years, we're likely to see some pretty significant changes. For example, with Aedes albopictus and with the amblyomma, the Lone Star tick, as things warm, eventually it's going to get to a point here where they'll be okay. You know, when that happens, though, I have to admit, I wonder what will happen with deer ticks. You know, give it 100 years, maybe it'll be too hot here. You just tend not to see very many deer ticks down in a place like Georgia. They behave really differently there, in Oklahoma, Texas, so you don't see anything like the amount of Lyme disease in the southern U.S., even though they have ticks there, the same species of ticks. They just don't behave the same way.
All right.
So thank you very much.